All right, so we left off talking about proteins and their structure. So originally we talked about our large molecules or organic compounds. We talked about carbohydrates already, which included your sugars. We talked about the monomer and the larger particles, or the larger polymer, if you will. Then we talked about lipids. We talked about a monomer of that. We talked about triglycerides. We talked about a phospholipid, okay, and steroid-based molecules as well. And then we started talking about proteins, which is what we ended with. And what did we say the primary structure of a protein was? It's just the sequence, the amino acids linked together by a covalent bond. That covalent bond had a special name because it's a protein. A peptide bond, which is just a covalent bond between two amino acids. And so just the sequence, the order of them and how they're linked up, kind of like the alphabet to make up words, was primary. Secondary structure was called what? We had two conformations, two shapes. An alpha helix and a beta pleated sheet. Okay? Those are due to what? So this primary sequence folds up on itself either as an alpha helix like a slinky or a kind of, you know, triangle, beta pleated sheet shape. And that's all due to hydrogen bonds. Okay? Then we said it folds up even more. So you might have more alpha helices or beta pleated sheets, or an alpha helix and a beta pleated sheet that will turn around and be able to hydrogen bond and some other bonds as well and create a tertiary bond. And that one tertiary structure can be one protein or one polypeptide. Okay? Then if we link more than one tertiary structure, so it's two or more, so we have a tertiary structure and a tertiary structure, and they bond to each other through hydrogen bonds or covalent bonds or ionic bonds or anything else. Now we have a quaternary structure, a fourth degree structure. Okay, so that's what we just barely ended with. So that was, we had one tertiary, oh, okay, hold on, draw. We had one tertiary, come on. There you go, one tertiary structure plus another tertiary structure to give you a quaternary structure. And one of the things that we talked about that could do that was, like hemoglobin, was 4 tertiary structures that came together that makes one hemoglobin molecule to be able to carry oxygen around your body. So that was that. And now we want to talk about fibrous proteins. So you could have a collagen type protein structure or a fibrous protein structure. So for collagen, its main structural protein is made out of collagen molecules. So we have main structural protein is collagen. And what do you think it is with elastin? Elastin. It's elastic fibers. So basically, with collagen, the main fibrous protein is collagen. Elastin is a protein that's part of the flexible elastic fibers. The difference between collagen and elastin, it's not so much that it can stretch, but that when you let go of the stretch, it can recoil back to its original shape better. So we call that recoil. And so let's write, it's good recoil. Okay, which is important. So elastin is the monomer that makes up the elastic fibers. It has recoil. It's not, elastic isn't built for strength. So collagen is more for the strength. So if we needed more of a strength, a tensile strength protein, we'd go with a collagen protein being made and use that. But when we need flexibility or stretch that allows recoil to come back to its initial shape, that's when we're going to look at using elastin fibers, okay, protein fibers. And let's see. And so then we also have globular proteins. Okay, so fibrous proteins, we're talking about the structural component. Globular protein, we're talking about the functionality of it. So an example of a globular protein might be hemoglobin. Okay, so these are proteins that do something. So hemoglobin transports oxygen. It does something for us. So we have other examples like antibodies. So these are proteins that function in immunity. Hormones are our chemical messengers. And enzymes are our biological catalysts. Okay, so an enzyme, we said, helps make a reaction more likely to occur or helps it speed up the reaction. So it's a catalyst. It doesn't get used in the reaction. It's not a reactant, and it's not part of the product at the end. It gets recycled. This comes in, it does its job, it goes away. Usually it's bringing reactants together and goes away. Grabs two more reactants, brings them together, goes away, and keeps doing it over and over again. So biological catalyst. And they make chemical reactions possible. So which of these three did we see in another category? Out of antibodies, hormones, or enzymes? No? Hormones. Hormones can be protein-based, but we learned that they could also be lipid-based, steroid hormones, right? So you could have a hormone that's made out of protein, or you could have a hormone that's made out of lipid or steroid. Then we get to something called protein denaturation or loss of conformation. So this is a loss of structure, okay? So if you're losing its structure, this could happen for a couple different reasons. Acid and heat will definitely cause this. We talked about if you had a fever, it could end up damaging your proteins, right? Or break down an enzyme so that it can't function if it was because an enzyme is a protein, right? So if the heat is too high, remember the example, you take an egg, you crack it, you put it in the pan. The protein part of that egg is the egg white part, which is the clear part when it's not cooked. But when you cook it, you add heat to it, it turns white. Okay, that's the protein that you've changed its conformation. It's not the same as what it was when it was the liquidy stuff. So acid or heat will usually break, and what it's breaking is the hydrogen bonds. So the polarity of it. So hydrogen bonds, we said, aren't super strong, but it allows for the things to weakly bond together. It's still an important bonding pattern, right? But they're easy to break. So we talked about fevers already, and let's see. So if you added just gentle heat to something, let's say, okay, here's a good example, hair, right? So I could take my hair, it's really straight. It's not curly at all. I could take my hair, I could take a curling iron, I could apply gentle heat, not like the girl that used the curling iron and then her hair fell off because she heated it too much. She destroyed the protein on that one. But if I took the curling iron and I applied gentle heat and I removed the curling iron, I have changed how my hair is sitting because I have heated the protein. I made new hydrogen bonds, technically, and it would stay that way until I took a shower and allowed my hair to go back to its natural conformation, which is not curly for me. If you had somebody that had curly hair, you could take their hair and you could straighten their hair out, and it would stay that way until, humidity or they wash their hair or something, and then their natural conformation would come back as long as they hadn't destroyed the protein completely. And the reason why can it go back to its original shape if you didn't destroy it? If you didn't destroy it, you still have primary structure. The order and sequence of the amino acids, then it will go into its secondary structure, tertiary structure, quaternary if it was more than one, right? So if you don't destroy the protein, you still have the same amino acid sequence. The bonds weren't broken. Those covalent bonds stayed the same. So gentle heat, not destroy it, like cleave it off and break it, but gentle, you could get your same conformation back when you're done. So as long as we don't completely destroy it, hydrogen bonds will form again. And that's not a problem. We'll get our shape back. So let's see. What do I want to tell you now? So like if I had applied heat, for example, so let's say this was I had an alpha helix with my pattern and I had my hydrogen bonds, and then I apply, we'll say, gentle heat, not superheat, and then maybe I have now caused, due to the heat, a different pattern of hydrogen bonding, so I changed the shape of it temporarily. And then I could wash my hair or heat it again or do something else. As long as I don't destroy that primary sequence, I didn't break that apart, then when it goes, you break these hydrogen bonds again, then it'll go back to its natural conformation. So as long as you don't completely destroy it. That's all we're saying there. But kind of like when you add enough heat in the fry pan with the egg, it doesn't go back to the egg white, not cooked part, because you have completely changed. You've added more heat than what would have allowed that to go back to its original conformation. Yes?
So you're destroying the protein structure when you cook an egg?
Yeah, you're breaking bonds. You're applying a lot of heat. Because you wouldn't stick your hand in the fry pan. That would really hurt. So it's not gentle heat anymore. But even some proteins are going to be a lot more sensitive. So you could have a low heat and it would still end up cooking it too much where you can't go back. So even if somebody does like a perm in their hair, right? So that structure change, they've actually done more than breaking and reforming hydrogen bonds, because they've used other chemicals to create more covalent bonds, and the covalent bonds are hard to break. That's why somebody who either completely straightens their hair or curls it with a permanent, meaning they're using other chemical means to do it, it'll be permanent until their hair grows out, right? So then they might have curly hair here and then it's straight up there where the new growth is. But it's because they created covalent bonds. It's not the hydrogen bonding then. So let's see, what else do we want to talk about? Enzymes and enzyme activity. All right, so we said it's a biological catalyst. All right, so globular proteins, they have a function to do something. It's not just the structure of it. So biological catalysts speed up chemical reactions. Okay, so speed up chemical reactions. And for example, this pen in my hand, okay, so because I have enzymes that function and do various metabolic processes within my body, I can lift this pen and it takes me 1 second to do it, okay? Versus if I had no enzymes to help me do this, it might take me 12 days to eventually get my hand to pull that pen and lift the weight of the pen all the way up to what I wanted it to do. So it speeds up a reaction. It could happen in 12 days, but I could speed it up with the use of enzymes. So I have this protein structure that can do it faster for me, okay? It basically just speeds up a reaction that would have occurred anyway. It's just going to take a lot longer without enzymes. And what else do I want to tell you? Enzymes are very specific. So you have one, and I usually abbreviate enzymes ENZ, one enzyme per one type of enzyme reaction. So for example, if I said A plus B, and I needed an enzyme to bring those two reactants together to get my product, AB, could I use the same enzyme for C plus D to get CD? No, because enzyme that would have recognized reactant A and B does not recognize a reactant C or D. They're different. So enzymes are very specific. They have one job, only one job. They can't work for other people. They can't make something else happen. Basically, there is a site on the protein, this enzyme protein, that recognizes something that fits in it. So it's a shape thing. Sometimes bonding occurs when they're doing it, but this is, again, the enzyme is not a reactant or a product. So if that shape isn't right, A, B, C, and D look different from each other. And this enzyme only fits what A and B look like. That's it. Nothing else. Won't work for C, D, E, F, G, H, I, J, K, L, M, N, P, Q, R, S, T, U, V, W, X, Y, Z. Okay? It won't work for anybody else, only for those. So if you have an A, but you have a C, it still wouldn't work for that either. It It will only work for the A and B together. And an enzyme that would work for C and D would be totally different. It won't work for A and B. So that's why we say one enzyme per one type of reaction. It's very specific. Enzymes are not consumed, so they're not changed by the reaction. Not changed by reaction that they work on. They're just recycled. Okay. Enzymes are always a protein. And that's why we said if you got a fever too high or something happened like that and it was way too high and it actually starts breaking the, not hydrogen bonds, but the actual other bonds of this protein that's your enzyme, and now you have no enzyme to make the reaction work, you are SOL. All right. Enzymes names usually end in the suffix ase. So remember on sugars, I said if it ends in ose, it kind of is a sugar, right? Glucose, fructose. It's telling you it's a sugar. If it's, well, lipids don't always follow that. Let's see. So enzymes ending in ase. So if you see something that's like dehydrogenase, it's some sort of enzyme, protein enzyme that will help break things apart. If it's, here's one, alcohol dehydrogenase. So if you did not have alcohol dehydrogenase and you drank alcohol, you would not be able to break it down. You'd get super goofy super fast. And some people don't have very much of that enzyme. So I'd be like a lightweight. I'd have a drink of something and I'd be giggling the whole time. Somebody else that has a lot of alcohol dehydrogenase has a lot of enzyme to break down the alcohol bonds, right? So that you can excrete it from your body, might have a hollow leg where they drink and drink and drink and they don't seem to get goofy at all, right? So ACE, you know it's an enzyme. If it's an enzyme, it's a protein. Let's see, what else? Enzymes functions, they are used to build or break down. So it could be either way, right? You could have A plus B, you're putting them together, you're building to get product AB. What kind of reaction would that be if I am building? It's a synthesis reaction. What's happening energy-wise? What's another name for that? Ender or exergonic? Endergonic. Okay. If it's an enzyme used to break something down, now what kind of reaction am I doing? Decomposition, or another name might be exergonic, because when you break a bond, you release energy. So make sure you guys play with that terminology. Use it as you learn new things as we're building on the topics and stuff. Let's see. So if we look at this picture down here, and so the same enzyme that brought A and B together to make AB, you'd have to have a different enzyme to break apart AB. So let's call that enzyme X would do that. That's really an X. Geez, this pen is not lining up very good there. Let's try that again. So we said that was enzyme X. Then you might need to do enzyme Y to do AB to break it down to an A plus B. And you might need enzyme Q to do C plus D to give you CD, but you'd need a whole nother enzyme like enzyme T to break down CD into C plus D. Because CD together does not look like a C plus a D, right? That's what we're saying. Think about it as a shape thing. So that's what the next picture is trying to show you here. So you have this blue blobby thing, which is a protein. It's an enzyme. You have, in this case, they're showing you glucose is they give you new names. So substrate is another name for reactant. So they're showing you if I bring in 2 glucose molecules and I have my enzyme, and then I use my enzyme to link a glucose molecule onto attaching it to, I'm making a bigger molecule, I'm synthesizing right? So that would be an endergonic reaction. It's taking energy in order to do this. I am building a molecule, so I'm probably using it for storage. So I'm going to store my glucose for later. We store it later as, if it's a plant, it would be starch. If it's a human, I'm storing it as glycogen. Okay, so I link my many glucose molecules together to make glycogen. So it's showing that the enzyme is bringing the one glucose and attaching it to my elongating glucose or glycogen chain, and then it releases the glycogen molecule or the glucose that I have now attached to it. And here is my enzyme that is ready to do it again. Okay, it's not a reactant, it's not a product. It's ready to grab the next glucose and link it onto another larger molecule. So that's the whole thing with that. If I needed to use an enzyme on a lipid type molecule, I could call it a lipase. Because remember, ACE is the enzyme, so it's saying this would be some sort of enzyme to break or build a fat type molecule, right? Protease should indicate to you it is some sort of enzyme that is going to work on breaking down or building a protein type molecule. Break protein bonds or peptide bonds, right? Carbohydrase would be the same thing but for carbohydrates. It's an enzyme, which is a protein used to break down or build carbohydrate type molecule, right? Any questions on ender, exergonic, all that fun stuff? So we are almost done with exam material 1 stuff. I will tell you when we reach the end of exam 1 material. Thursday, so next class will be a week before the exam. And so on the A&P website, Sherry will activate what's called the learning pathway. And so right now it just shows like exam 5 material from the end of last semester. But on Thursday it'll get activated, so you'll see the study guide for exam 1 and everything that we'll cover topic-wise. And then you're also going to see the learning pathway is set up where you have to go through the pathway or the steps, follow the breadcrumb trail. So it says, do this first, and then it allows you to move on to the next step. Now do this. And what I would suggest is that that if you are already in tutoring, that if your tutor is saying, explaining a concept to you, I want you to say to them, okay, wait, let me make sure I've got that. And I want you to re-explain it back to the tutor or if you're in a small group and you're working with other students, explain it back to them, because it's when you verbalize it that it starts to make sense and you catch where the holes are in your thinking process. This class, sometimes the material I'm telling you, it seems like, oh yeah, I got that, no problem. And you have a general idea of what I've just talked about, but you may not remember all the itty bitty details. And I guarantee the exam is going to test you on your itty bitty details. Okay, so what I suggest you do, once you get that pathway going, when the exam 1, not exam 5, but the exam 1 study guide comes up, it's just a bunch of sentences saying, make sure you know how to do this. Make sure you know endergonic versus exergonic. Make sure you understand homeostatic imbalance and yada, yada. You should take that and get with your small group of buddies or your tutoring session and ask each other questions. Have your notes in front of you, quiz each other back and forth, see if you understand the concepts and what happens with positive feedback versus negative feedback, and give examples to each other and things like that. And then you will also have the option to look at old exams. They're from a bunch of years ago, but if you take the exam and you just use that as your study guide, you will not do well in the class. And the reason I say that is because most people will take an exam and they will just look at it and they'll just go, all right, question 1, here's the right answer. Question 2, that's the right answer. Question 3, and I will tell you that if you think you're just going to memorize that or you've got the concepts because you got it right on there, that when you come in and take the exam in here, you're going to be like, oh my gosh, it's all jumbled up. It all looks the same. I don't know which one is which. And so if you really want to learn how to study from an exam, you take the exam and you go through and you figure out what is right and what is wrong about every single answer choice. So you make a wrong answer a right answer, you make a right answer a wrong answer, you change the question itself to see if that would change how you would have answered it down below. That's how you know if you know the topic inside and out. And have your material next to you, your notes, so that you can look up the answers. That's a form of studying, having to go back and look up your answers and stuff. Okay, that would be the best way to study. And I will tell you now, I have had students that have gotten a 40 or a 60 in the past on their first exam. And some of them will continue to get the same exam score all the way through. And then I've had some students that you tell them, these are some good study skills, try this instead. They will get that 40 or 60 maybe the first time. They change what they do and they have pulled an A by the end of the semester because they got an A on all the other exams. It is possible. You just have to learn how to study effectively and efficiently instead of spinning your wheels and not, feeling like you're studying a lot, but you're not getting a good grade. Okay, so that's my spiel on that. It'll start popping up next week. Internet students will see something come up on their Blackboard account that will tell them to sign up for the exam. You guys will take the exam on Thursday, not this week, next week, Thursday, 1 to 2:15 p.m. If you get done early, you leave. You don't have to stay the whole time. Okay, and room 106, Meyer Health Science. Cool? All right, let's move on. So, continuing on, this material is still on exam 1. I will let you know when we are done with that. But let's look at nucleic acids. So here's another big molecule. And what we're talking about is DNA and RNA. Anybody know what DNA stands for? Deoxyribonucleic acid. And RNA is ribonucleic acid. Deoxyribo and ribo. The deoxy and the ribo part, they're talking about a sugar molecule, ribose, whether it has an oxygen on it or not. That's pretty much the only difference there. So let's see. So the nucleotides are the building blocks. Those are the monomers of a DNA or RNA molecule. Okay? And the nucleotides, there are 5 of them. So we have adenine, we have guanine, we have cytosine, we have thymine. Those 4 are all found in DNA. And the key there is both DNA and RNA have adenine, guanine, and cytosine. The difference is RNA has a uracil as its nucleotide instead of thymine. So if you see uracil, you know you're talking about RNA. If you see thymine, you know you're talking about DNA. Okay? And let's see. Do you guys want me to spell out the adenine, guanine? Yeah? Okay. So you have adenine, we have guanine, we have cytosine, we have thymine if it's DNA, or uracil if it's RNA. All right, let's see. So those are the nucleotides. These molecules here are your monomer. We link these monomers together, and then we end up getting our polymer, if you will, which would be the DNA molecule or the RNA molecule. So 2 classes of nucleic acids, DNA or RNA. DNA is the genetic material. It has our genes on it for the cell, okay? So this is what's on your chromosomes. You could think of DNA, which is housed in the nucleus of a cell. You could think of it as like the Joy of Cooking cookbook. It has all the possible recipes that you need to make anything on your body. Okay? How to make your eyeballs, eyeball color, hair color, curly or straight, skin tone, height, how to make a pancreas, how to make your lung, how to make the enzymes to make something function so that you could do your other metabolic processes, so on and so forth. So DNA is all of the possible recipes. You don't necessarily cook all the recipes in your cookbook, right? Maybe some of them you don't like, maybe some are redundant, I don't know, but you don't necessarily make all of them all the time. But the ones that you do want to make, you pull out your recipe book and you open up to the one section that you want to make. And then you can take that one recipe and look at it and read off just that one little thing. Maybe you make a copy, like a little recipe card, so you don't have to keep opening up the big book. Okay? That little recipe card is going to be like RNA. It's like the one little section that we need to read right now, and that's it. Okay? We make RNA because we don't want to damage our DNA. So this is for safety purposes for one thing, right? And also DNA does not leave the nucleus. DNA stays in the nucleus, but RNA is made in the nucleus and then leaves the nucleus and will go out and find a ribosome. The ribosome will read the RNA. The RNA is the message, the little recipe card. Ribosomes will read that and make a protein. That protein may be an enzyme for another reaction later on. That protein might be a collagen protein that you need for your hair or your nails or your skin, so on and so forth, okay? So that's where we're going with this whole DNA, RNA thing. So DNA is the genetic material for the genes. It's the blueprint for the body. It must not be damaged or mistakes can occur. So for example, I mentioned this on an earlier lecture when I said sickle cell anemia, okay? So instead of the red blood cell having its little concave little look to it and being able to carry oxygen properly, it gets a sickle shape to it, okay? And then it can't carry oxygen efficiently or effectively, okay? And so sickle cell anemia is due to one single nucleotide mistake, one. So maybe instead of an adenine, it put in a guanine instead. And that's it. One little mistake, you don't get the right protein. And then something won't have the right shape or function to it. Sometimes it won't function at all. Sometimes it has some functionality, but not nearly as good as what it should have been. So it just kind of, rarely, occasionally, it might actually function better, like superhero X-Men kind of thing, but probably not. Seriously. Okay, so they have a picture down here. Let's see, what else? Location, so I said DNA is found in the nucleus of the cell. The DNA structure is also that it is a double strand of nucleotides. So this is another difference between DNA and RNA. DNA is a double-stranded molecule, so it's two strands of nucleotides that create the double helix and kind of twist up on itself. RNA is a single strand only. So later when you're trying to review and study this material, make a chart. DNA on one side, RNA on another. All the things that make up DNA, all the things that are either the same or different about RNA. So compare and contrast them. That'll help you kind of get them under your belt and realize which one does which. Where do you find them? What does its function do? Single strand or double strand? Uracil or thiamine? You know, that kind of thing, right? So let's see. And the nucleus of the cell is your control center, right? So we have it nicely protected in the nucleus. And we said it's double-stranded. And so, for example, if I were to write out a DNA structure, let's say we have A, T, C, G, C, T, A, C. So what kind of bond are between the nucleotides? A single covalent bond, right? So is that a strong or a weak bond? It's strong. It's not going to just break easily. This would take some work if we want it, but we don't want to chop this up. We want to keep this intact, right? So it pairs the other strand, so that's one strand. It pairs, and certain ones pair up with each other. So what you end up finding is you have a pyrimidine pairs up with purines, always. And if you look at the shape, so a pyrimidine is smaller. So I think big word, smaller molecule, okay, or smaller structure, single one. Purine was the shorter word, but it's actually the double ring structure. Okay, so a single ring is the long word, pyrimidine. Double, you don't have to know the actual structures of adenine, guanine, cytosine, and thymine, and uracil, but you do need to know that a purine attaches to a pyrimidine. They pair up, they match up, this is their buddy, okay? So if we look at the structure of DNA, we had, here's that sugar group we talked about, ribose sugar. And the reason it has here OH and RNA or H and DNA, because DNA I said is deoxy. D means like take away one less oxygen. So they're saying it's just an H in that square if it were DNA, but it would be an OH or a hydroxyl group if it were an RNA molecule. So deoxy removed an oxygen. Ribonucleic acid has the OH in it because it has the oxygen. That's the only difference. So the sugar could be different between a DNA or an RNA molecule. The phosphate group is the same. which is polar. And then depending on which nucleic acid it is, you will either have the pyrimidine or the purine attached there. So it's off of the 1 carbon that you have, so we call that 1 prime. The 1 carbon has the base attached to it, purine or pyrimidine, which would either be cytosine, uracil, thymine, adenine, or guanine, depending on your structure. So for example, if I said that this is a ribose sugar, okay, with the oxygen on it, would it be a thymine or a uracil? If it has the oxygen, it'd have to be uracil, correct? Because if it were thymine, it would have to be the deoxyribose. So look at it a couple different ways. Phosphate group doesn't change. That's off the 4 carbon on the ring on the sugar. The 1 carbon is the one determining which nitrogenous base, cytosine, uracil, thymine, adenine, or guanine is attached to it. And then it's off the 2nd carbon is what determines whether it's the ribose or the deoxyribose. Okay, so what else do I want to tell you? So the other thing is A's always pair up with T's if it's DNA. So A's and T's pair up together. Sorry, don't do a straight line. That would be a covalent bond. Guess what kind of bonding brings the 2 strands together? Hydrogen bonds. So A will always bond with a T if it's DNA, or if it were RNA just binds to the DNA, one side of the DNA molecule for a short time. So A's are going to be either a T or a U. But remember, RNA is not a double strand, so it's just temporary when it bonds. That's why we don't talk about it a whole lot. And C's always pair with G's. G's always pair with C's, T's always pair with A's, and vice versa. So if I wanted to draw the other side that will hydrogen bond to this side and complementary base pair with these guys, who would I draw under the A here? A T, and then who would be on the next? A, G, C, G, A, T, G. Okay, and they would have hydrogen bonds between them, holding these 2 strands. So that's what we mean by double-stranded. Okay. And what else do I want to tell you there? It's a complementary strand. So if you know one strand, you can always figure out what the other strand is, no matter what. And we talked about hydrogen bonds. And we're going to end up talking about, with RNA, there are 3 types of RNA that we'll mention in a little bit. The main one that we'll talk about first is messenger RNA, so we usually write it as little m, capital RNA. Here is a picture of just RNA, so that's why it's single-stranded. It has the phosphate is on the backbone side of the molecule, okay? And then it has the base pairs hanging off. So here is your ribose sugar, here's your phosphate group, and then it's the ribose and the phosphate that make up the backbone of the RNA structure. On the DNA structure, it would be the deoxyribose sugar, so I'll put D-ribose, and the phosphate that make up its background. So it's the sugar and phosphate that make up the backbone, or the sides of the ladder rung if you want, and it's the base pairing that hydrogen bonds to the other side of the base that makes up like the ladder rungs. Okay, that's why they're showing you the hydrogen bonds there. What else do I want to tell you? So basically, you could take the nucleus out of a cell, and all cells have a nucleus except for one cell. Anybody want to take a guess? Red blood cells don't have a nucleus. They originally do, and then it gets taken out. So for our purposes, red blood cells, no nucleus. So if you took the nucleus and stretched it out, you would find the DNA would be about 6 feet long inside that nucleus. That's pretty cool. You guys have started to look at some cells yet in the microscope, or is that going to be this week? So look at that nucleus. 6 feet of DNA inside of there, in that nucleus. Not even the whole cell, just the nucleus of the cell. And that's why it has to compact and twist up and do its funky shape and stuff too. What else do I want to tell you? And the nucleus of each cell has all of the blueprints, the Joya Cooking Cookbook, if you will, in each cell. So every cell knew how to make any other kind of cell in your body. But we kind of turn and turn off things later so that we can't just do it. That's why people are so interested in stem cell, because stem cells can be turned into any other cell. But once you've kind of designated this cell to be a heart cell, and this cell to be a muscle cell, and this cell to be a lung cell, it kind of only stays that way, right? But it has the information to make another cell. Most organs can't, like your brain can't, heart can't. But most of your cells can make another identical cell so that you don't look like Swiss cheese later when one cell dies. And some cells live longer than others. They have a time limit. So like your liver cell, they kind of regenerate about every 6 months or so. So you can drink a lot and then 6 months later drink a lot more. I'm kidding. Don't drink a lot. But anyway, so what else do I want to tell you? So what do you think cancer is? Cancer may be damage to that DNA, the original structure, if it keeps saying, keep making more, don't turn on the off switch, right? So skin cancer, UV light could change the DNA of a cell and cause it to now have a mistake in it, and it keeps making that mistake every time it replicates now. So one thing to look at. Let's see, nucleotides are covalent bonds. Two strands are held together by what kind of bond? Hydrogen bonds. So in order to read our DNA, we have to open it up. So we end up having to say, wait a second, I need to read the gene between this area and this area on the DNA. I just need that one gene. So I'll have various enzymes that can come in, unzip that one little section, read it off. This is where the RNA comes into play, where RNA will go, I'm that one little message, but make a copy of the DNA. I'll be RNA copy. So that way you don't damage DNA. So this is the one little piece that we will read that off and make an mRNA molecule from it, the messenger RNA. And once we're done reading off that message, that DNA will zip back up. It will re-hydrogen bond with the other side, its complementary base pair, and zip itself back up and go back into its double helix shape in there. So have you ever had a zipper where like it opened up in the center for a second? That's like what we're doing. We're opening up one little section, but this time our zipper works and it zips it back up instead of now it's stuck and it won't work, right? You know, never mind. Okay, so if we look at RNA now, so RNA has adenine, uracil, guanine, and cytosine. So you're not going to find any thymines in an RNA structure. They don't belong there, okay? This is a photocopy of the gene in the DNA, that one little message, the one little piece we decided to read off. So that means that this has to complementary base pair to one side of that DNA strand just for that short period of time. So that's why I was saying, if we go back up to here, that if let's say this is our DNA piece that we unzipped, and let's say it's connected to the rest of the DNA over here, right? But we unzip this one section and we need to read off that one gene. So what would the piece of RNA look like that base pairs with that one section of DNA? So an A would be a U, a T would be a A, this would still be G, C, G, A, U, G. And then this piece doesn't have, it only shortly hydrogen bonds with that side just while it's being made. And then those hydrogen bonds will break off and this strand, this single strand, will be the mRNA or the RNA molecule that we're going to talk about. The DNA would zip itself back up and look like that upper one that we did the first time. Okay? So, photocopy of the genes in DNA is what makes our mRNA molecule. This is used so that the original master copy, the DNA, is not accidentally damaged, okay? So used to keep, that's a K, DNA protected, thus not damaged. Okay, and RNA is made in the nucleus because DNA can't leave there, and we had to make it from the DNA first, right? But it will then leave the nucleus, and where we find RNA hanging out is in the cytoplasm. So that's why it says location is synthesized in the nucleus, then transported to the cytoplasm. So RNA doesn't hang out in the nucleus, it gets made there and leaves. And the structure we said is a single strand of nucleotides. So what do you think a nuclease would do? Nuclease. ACE means it's a enzyme. Nuclease. It is some sort of protein, enzyme protein, that either breaks down or builds nucleotides. So it's either going to link them together or break them apart, depending on what its purpose is. So look at the base of the word, try to figure it out. If you see ACE, you know it's an enzyme. And let's see, what else do I want to tell you? It's always made in the nucleus, transported to the cytoplasm. So it's in the cytoplasm that we will find nucleases. So in cytoplasm is where we will find nucleases. There's your ACE. So in this case, nucleases break down nucleic acid. DNA is protected in the nucleus because it doesn't see these nucleases ever. So we don't have to worry about our DNA getting damaged by a nuclease by accident because nucleases are only in the cytoplasm. DNA is not in the cytoplasm ever. So let's see, when we open up DNA, we make mRNA, we are expressing the gene. So it's called gene expression if you've had some of the other biology classes. And we don't need any more expression of the gene. DNA then closes back up. And as long as we need the gene expressed, DNA will stay open, making the mRNA. So we could leave that one little part of DNA unzipped for a little bit and keep reading off mRNA over and over again, depending on how much we need to express and make these other proteins. Okay, or we say, yep, we don't need any more of that, close it back up, zip it back up, and it won't read off any more mRNA for a while. Okay, and then after a while, those RNAs will get chewed up by those nucleases in the cytoplasm because we won't need it anymore. And then when we say, wait, we need more protein, we need more of those messages read off, we can go back, open up the DNA, read off another mRNA that will keep reading and doing its job until it gets chewed up by nucleases in the cytoplasm. So it just, depending on what the body needs, we don't want to make stuff more than we need to, right? Why waste energy? Why make proteins that we don't need right now? So it just depends. What is the need for that organism or body at that time? We can make it, we can stop it, we can make it again. We can keep making it until we don't need it anymore, so on and so forth. Would that be like negative or positive feedback? Maybe. Think about it. All right, so let's see. We already talked about the difference between RNA and DNA and how you can tell the difference. So for example, let's say, like deciding on whether you're going to keep reading off a message or not. So let's say you just ate a meal. So DNA for insulin, the gene on the DNA strand for insulin is opened up, and we say we need to make insulin, okay? So we open it up, messenger RNA is made from the template strand, template means original strand from the DNA, for insulin. And we will continue to make this messenger RNA as long as insulin's in the blood, in the bloodstream, and it remains high. Once the insulin levels start decreasing, okay, the gene closes up and we stop making messenger RNA. And after we make mRNA, it leaves the nucleus, goes to the cytoplasm through these things called nuclear pores, which you're going to see in the next chapter. And it looks for a ribosome, which is anytime you hear the term ribosome, you know it's a protein factory. Ribosome's job is making protein. And sometimes we have RER, rough endoplasmic reticulum. It means it's endoplasmic reticulum that has ribosomes on it. And sometimes we have free-floating ribosomes. But if you see a ribosome, its job is what? Make protein. All right, you got it. Ribosomes latch onto the messenger RNA, they read it, and then the nucleases try to find the RNA, but as long as the ribosome is attached to that messenger RNA to that message, the nucleases can't eat away at it, okay? Once it's done being read off in the ribosome, a nuclease might be able to find it and start chewing it up. That's why we got to keep making the messenger RNA or keep making the protein, whichever happens first, depending on what's going on. And so once the RNA is no longer protected, so as long as insulin is high, another ribosome attaches on many messenger RNAs are present at once until the insulin decreases. Then the gene closed, so it stops making the mRNA, the nucleases overtake the messenger RNA because now there's less mRNA being put out there. So it's kind of like, you know, how much do you have more of? Now you have tons of nucleases that can chew it up. You have less mRNA being made, so it's more likely to find it. And you no longer need the protein that the messenger RNA was coded for, so the nucleases break down the rest of the mRNAs. And you don't need to do it again or unzip the gene for insulin until the next meal, for example. So that's why, you know, make it when you need it, stop when you don't need it, right? So if we look at another molecule called adenosine triphosphate, I'm going to put some spaces here so I can bring it up on the page. Okay, so ATP, adenosine, what does that sound like? Adenine, maybe? Look, it is adenine, the nitrogenous base. Is this a purine or pyrimidine? It's a purine. Remember, short word, double ring. Long word, pyrimidine, think single ring, shorter. Just think opposites on that one, I know. It has a ribose sugar. We just saw that. Also attached to something else. But instead of one single phosphate, it actually has 3 phosphates attached to it. And these phosphates are highly negatively charged. They've got a lot of oxygens, a lot of negative charge to it, right? Lots of electrons. So it's kind of like having siblings that really don't like each other. And you make them hold hands. Be good, hold hands, right? But they don't really want to hang on to each other. But they're being forced to by their parents. So it's an explosive situation, right? Because they're fighting and they don't like each other. So they're kind of punching each other and trying to get away. And so it's really explosive when they finally, you know, Red Rover, and they break the bonds and they separate. A lot of energy gets released, right? Kids start running wild. That's what you're kind of thinking here. These are high energy bonds between the 3 phosphates. Okay, so basically, I want to tell you. So if I were to write this reaction out, you have ATP. T is for triphosphate, so it means 3. That's the 3 phosphates. So ADP is adenosine diphosphate. That would mean there's only 2 phosphates. AMP, we don't really talk about in this class, but AMP, adenosine monophosphate, means there's 1 phosphate. So we can add and take off phosphates as we need to. This energy molecule gets recycled constantly in our body. This is important for us to have energy. So ATP can be broken down into ADP plus a phosphate, right? Because one of them got broken off. And you could do ADP plus a phosphate to get ATP again. And you'll see me, I typically write a phosphate like a P with the circle around it because it's kind of encompassing. When we put the P, it really still also means you have these other 3 oxygens on there. Okay, what else do I want to tell you? The squiggly lines just mean a high energy phosphate bond can be broken to release tons of energy. Lots of energy when we break the bond. We need this kind of energy for almost all cellular activities. And again, we have adenine, ribose, and 3 phosphates that make up your ATP molecule. All right, we're still in exam 1. Let's jump to biology of the cell real quick. We'll just do a quick little overview of this, and then we'll pick this back up when we come to lecture on Thursday. But so this is a cartoon that helps us understand the various parts of the cell and their function. So it's a representation. Not all cells look identical to this. If you guys did get a chance to look at a cell last week, if not, you'll see it this week. But the main thing that you guys can pretty much only see when you're looking under the microscope is you see one big globular, circular, dense-looking thing, and that's the nucleus. And then you typically can kind of see the outline of the cell membrane, okay? The cell membrane is made out of a phospholipid bilayer. So this is where we were talking about phospholipids before. So technically on this phospholipid bilayer, if I took out a chunk of it here, it would be the hydrophilic heads, right? The squiggly tails are the hydrophobic hydrocarbon chain, right? Then on the other side, same thing. Hydrophobic chain, 2 fatty acids in the center with the hydrophilic or polar head, okay? So that would be like this little edge right here is made, if we blew it up, looks like this. So outside would have water, inside also has water, right? You have tons of cytoplasm inside of your cell, okay? Then we have various things within our cell. What else do I want to tell you? Let's see. We're going to learn a lot about the cell membrane, how it works, what it lets into the cell and out of the cell, like nutrients in, waste out, just like us, but on a microscopic or smaller level. Let's see. So the plasma membrane was important for whether it lets things in or out. Okay. Let's see, what else do I want to tell you? How about these microvilli? So that's these guys right here. So can you see those little projections? All it is it's the plasma membrane that has these infoldings in and out. What do you think that does to the surface area of the cell? It increases the surface area. If you have more surface area, you have more ability to allow things in or out of the cell, more things to interact with you, okay? Let's see, what else do we have here? How about, we have mitochondria. They kind of look like a little weird kidney bean looking thing. So that's this guy here, mitochondria. So anybody ever remember the magic school bus? Powerhouse of the cell is the mitochondria, the mighty mitochondria. It's true. This is where you, especially in 228, learn the details of how you make tons of ATP through the electron transport chain, oxidative and reduction reactions. You're going to see this again in 228. This is why I'm bringing it up now. But this is a double membrane. This is where you make all the ATP that your cell pretty much, not all of it, but it's the main area where you make it. So that's why it is the powerhouse of your cell. That's why it's important. We also have, how about, depending on what type of cell it is, what its function for the cell is, what is its job, would depend on how much mitochondria you might actually have in a cell. So if this is a skin cell, let's say, it doesn't need tons of mitochondria in it. But let's say it's a muscle cell. Your muscles need to do work, right? You need energy to be able to do work. Or a heart cell. Your heart can't stop, or you stop, right? You need tons of mitochondria in those. So those cells will have tons of mitochondria in it. This picture is just showing you like 2 in it. So keep in mind, this is like, that's why we say general cartoony cell. Depending on what its function is, depends on what will be in it and how much of something in it. How about, what do you guys think? All right, we talked about a nucleus already. So here, the little blueberry in the center is the nucleolus. It has, then you have here the nucleus itself is the bigger part of the blueberry. And notice how it has like a dotted edge to it. Those little circle holes or spaces in between the membrane are nuclear pores. So I said DNA is kept in here. It doesn't leak out, okay? RNA is made in there, but it will leave through the nuclear pores to get out into the cytoplasm of the cell, okay? So that's how it exits that area. And that's called a nuclear envelope, the part without the holes in it. The holes are the nuclear pores in the nuclear envelope. And we also have, let's see, what else do I have? So the nucleus is typically centrally located, but not always. So like when you guys look at adipose cells, the nucleus is actually mushed up against the side of the membrane. so it doesn't look like you have a nucleus in an adipose cell. But most cells, the nucleus is kind of somewhat centrally located, which makes sense. Let's see. Nucleolus. Active area of RNA formation is where the nucleolus will be. Let's see. We also have, I'm trying to find where they say it on here. So we have this kind of pancakey thing here, right? So these are also made out of, it's membranous. So it's a phospholipid membrane also. And this is where we have our rough endoplasmic reticulum if it has dots on it. Those would be, the dots would be the ribosomes. So if you see a dotted thing, it's a rough endoplasmic reticulum. On here, they didn't really show it. I think they're showing it down here. If it doesn't have any ribosomes on it, it would be a smooth endoplasmic reticulum. So smooth endoplasmic reticulum, would it do any type of protein manufacturing? No, because I said ribosomes do that. So rough endoplasmic reticulum helps make proteins, but smooth has a different job. Let's see. So smooth, it makes more plasma membrane and lipids, and it's made out of lipids also on the plasma membrane. Rough endoplasmic reticulum, they are close to the nucleus, both RER and SER, and they are making products for exportation. So they're making stuff here, and everything then gets processed and packaged or repackaged, ends up going to this other pancake looking thing that's green, and that's called a Golgi apparatus. Okay. The Golgi apparatus is a processing plant, kind of like a winery. So winery is like the ribosomes making the protein, but they need someone else to package it and bottle it up and send it to the consumer. So that'd be your Golgi. Or you could think of it like UPS. They get this package and they're now going to deliver it, repackage it maybe in their box and send it on to somebody else. So that's the Golgi apparatus. And it's a one direction type of movement of stuff. So things are moving this way, going to the Golgi. Golgi is going to process it and move it this way. And it's kind of hard to see on this picture, but it ends up making a vesicle that comes off. So the packaged material is in now this green ball, which it's not really green, it's just color coding it you for cartoon. But it then will move either to something else within the cell or go out towards the plasma membrane. All of these are made with a phospholipid bilayer. So the membrane of your outer membrane changes. Kind of, I was saying, like, depending on the fats that you ingest in your body, eventually will get incorporated into your cell membranes. This is the same idea. Things that get processed and made end up merging. And so if you had, we'll talk about this next time, but basically if this was the side of the membrane, this is out, this is inside of your cell, and you had your Golgi apparatus, and then it ends up packaging something in here, and then this kind of breaks off with your material inside, and it ends up merging here. And then what will end up happening is it'll look like this with your contents in it that can now get to the outside of the cell. Okay? And what was the side here of the membrane is this, and then eventually it looks like this, right? And if I were to draw another arrow, then you have your membrane of your cell and your contents are now outside of the cell. So if we're getting it out of the cell, we'll call that exocytosis. We'll talk about this again next time. Exocytosis. If we're bringing something into the cell, it might be called endocytosis. So we can have a similar process where we have food or drink or something that we need to bring inside of our cell. We have our cell membrane, and then it will kind of do what's called like an invagination, where the material that was outside of the cell is coming in, and then it almost closes off, right? And then eventually it does close off, and now you can bring this into, I didn't mean to make it a smiley face, but there you go. And now you can merge this with some other vesicle called a lysosome, and it can attach to that one, and then they merge. The lysosome spills its enzymes, its digestive enzymes, into that vesicle that came into your cell, and it can break down that food, drink, water, whatever it is. So one way to remember another name, food eating, is endocytosis, bringing it in. Food drinking is pinocytosis, like Pinot wine. So think Pinot Grigio, you're drinking the wine, you're drinking it into the cell. So different terms for various things. What else do I want to tell you super quickly? Okay, so we talked about endo and exocytosis, and let's see. So a vesicle that they're showing you there, there's like a little green ball that they're saying, that's just something that could transport things in and out. And there's almost like a roadway inside of the cell, various fibers that it kind of can follow along. And those might be things that are made up of your, let's see, where are they on here? I don't know if they're listing it. So basically, like the various roadways and stuff, where do I want to find you? Microfilaments, microtubules, intermediate filaments, things that are found inside of the cytoplasm might be like almost like a string that the little vesicle can follow along on its pathway to know where to go to and from inside of the cell. And those things also help give shape to the cell, also a little structure on the inside of it. And let's see, you also have things called, so aside from microvilli, which help with the surface area, we also have things like cilia and flagella. They are different than microvilli. Cilia help move things across the cell's surface. So you guys will see when you're looking at cells this week under the microscope that some things will have these little hair-like projections on it called cilia. And a lot of times they move in one direction. So it's kind of like they will push things one way and then recock, and then push things and recock. And then you also have flagella, so that would actually move the whole cell. You guys all know at least one cell that has a flagella on it, right? Like a sperm cell has to get somewhere, right? So characteristics, so basic structural functional unit of life is the cell. That is the basic structural unit. Okay, let's see. The diameter is too small to see by the naked eye, thus we have to do what? Magnify it and look at it with a microscope. Magnify it. There are trillions of cells, okay, and in over 250 basic types of them. We're not going to make you learn all of that, right? But for example, they vary greatly in shape and in size. And so, for example, what do I want to tell you? So we have, like, here is an example of a muscle cell. Here is a red blood cell, and here is a nerve cell, right? They look very different from each other. And at first, sometimes when you guys are in lab and you're looking at all these cells, it all looks like gobbledygook to you. You're like, I can't tell the difference between this and that. This looks like the same tissue as that one. I don't understand. So look at what makes them similar to each other and then look for the differences. That will help you understand how to look at this cell and why you know it's this tissue type and not that one. These three all look very different from each other, right? So, for example, this should be a skeletal muscle cell because, one, it has striations in it. I can see some striping. It's multinucleated, so each of these is a nucleus. What? Some cells have more than one nucleus? Yes, yes, they can. Let's see. On the nerve cell, you have a cell body. So the main body of the cell is there. You have an axon, which is along that area, and then you have a process sticking off on the side. Some nerve cells, okay, are as long as your leg, actually a little longer. How about your sciatic nerve? One nerve cell, sciatic nerve, there you go. So some cells are really long or big, right? And some not so much. So general structure, you got the nucleus, the control center of the cell. Okay, so this is typically centrally located, we said. And what else do I want to tell you? The cytoplasm, or plasma membrane, outer limiting boundary of the cell. So that was our phospholipid bilayer, right? So this controls what goes in or out of the cell. Okay, this will make sense later when we're talking about diffusion and things like that also. And then with cytoplasm, general term for everything in between the plasma membrane and the nucleus. So it's kind of like a soup. It's a little slimy. Typically looks kind of pinkish due to the stain and sometimes blue depending on what they're trying to show you. They'll use different staining techniques on the slides that you're looking at. And it's like if you think of it like a soup, it's got a lot of veggies floating in it, right? Like a veggie soup. So that's kind of what you're seeing within the cell. You have this cytoplasm that has stuff floating around within it. Anything between the plasma membrane and the nucleus is going to be cytoplasm because it's not so much the fibers and the other stuff you're seeing in it. Those are the veggies floating around in it, but it fills up that entire space. Cytosol is the intracellular fluid. Intra is like within, intracellular fluid that makes up the foundation of cytoplasm. Okay, organelles are structures that perform specific functions. Okay, so small complex structures that have specific functions. We can't see these with the microscope. You would actually need something more, what's called an electron microscope. So what they do is they take electrons and they bombard this, and it looks like a very grainy like picture. And that's how you can tell it's an electron microscope picture. And inclusions are clusters of a single type of molecule. Let's see, I think we're going to save plasma membrane, also called cell membrane, for next time. And we will stop here for a moment. Implement that stuff that I talked about with studying. Start studying. You have most of exam 1 material. We're still on exam 1 on Thursday. But I just want you guys to stay on top of this. Don't get behind. Let's pause this.